to be detected by who? The hyper, the increase, or the decrease is going to be detected by the the receptor. So how many types of receptors did we mention? We mentioned there are photo receptors. Excellent. There are chemo receptors. In addition, we have thermoreceptors, photoreceptors, chemoreceptors, thermoreceptors. What else? Mechanoreceptors. So all of these receptors, and osmoreceptors for the osmolarity, osmoreceptors. So these receptors, they are the ones who are going to tell we have too much of something or too little of something, which are both bad. As we mentioned, we don't want, is not a situation of the more the better. We don't want too much of anything. We don't want too little of anything. We want just the right balance. Okay, so let's now assume we have a situation where there is a hyper or a hypo. So there is a hypertension, meaning that what? What does hypertension mean? Hypertension, from the word hypertension, what does that mean? That means very high blood pressure. Where? In the heart, or running through the blood vessels, slamming the walls, hitting the walls of the blood vessels. How do we know if there's high blood pressure? Do we put the blood pressure cuff on the heart or around the blood vessels? Yeah, so we know there's hypertension when the blood is running so hard and hitting the walls of the blood vessels, the tubes, hitting the walls of the tubes of these blood vessels. So those tubes are going to get dilated. And once they get dilated, they are going to be, the mechanoreceptors around them are going to be stretched out, going to be stretched out. And when they get stretched out, then what are we going to send? We're going to send a input. There's going to be an input signal, and we said input is always what? An input signal is always what? Is always sensory. An input signal is always sensory. And where is the input signal going to go to? You know, if you see something wrong, you're going to call 911. If you want the ambulance, if she wants the police, and if he wants the fire truck, the three of you are going to call 911. But it's the job of the 911 center to send the fire truck to the right place, the police to the right place, and the ambulance to the right place. So your brain is like the 911 center. It's receiving so many different sensations. One sensation, it's too hot, brain. Another sensation, it's too much calcium. Another sensation, blood pressure is too low. So your brain is the call center. It's receiving so many different sensory stimuli, but it has to generate a correcting what's going to be coming out. So if the input sensory signal, where is the input sensory signal going to get to? What's the 911 center called? Yes. Excellent. So that is the integration. That's the integration, processing, integration, processing, and control center. In this case, it happens to be the brain. Remember last week I mentioned the brain is not the only center that is responsible for processing and integration and correction. Who else is responsible? Who else is an example of a integration center? The pancreas. Remember we said when there's too much glucose, the pancreas acts out on its own to repair the problem by secreting insulin to bring down this too much glucose, this hyperglycemia. So if the integration receives a sensory input, what are we going to be generating? We're going to be generating an output. If the, remember, we said the sensor is a receptor. So the sensor, is it going to be sending out a sensory input or a motor output? No, you're not paying attention. The question is, if the receptor is a sensor, is the receptor going to be sending out a sensory input or a motor output? Yeah, a sensory input. Then the integration, the processing center, who many times happens to be the brain, but not the only one who's responsible for integration, processing, and control. What is the effect? What is the processing center going to be sending out? Motor output. So the output is always motor. The output is always motor. There's something that you guys learned last semester in A&P 1, and that is all sensory input is what? Is afferent. Is afferent. All sensory input is afferent. All input is sensory and afferent, while all output is motor and efferent. Very good. Is motor and efferent. And now that's a hint. Where do you think the output motor efferent stimulation heading to? You see the word if, effector. So it's going to go to the effector. And we said the effector is one of two. You know, when you call 911, the effector is either a police car or a fire truck or an ambulance, because they're going to do the effect. But in our loop here, the effector is not a police. What is the effector? Effector is one of two things. What is it? We talked about them. The sweat gland. Remember, when it's too hot, you sweat. And when it's too cold, what do your muscle organs do? You shiver. So our effector is always one of two. It's either a, no. If afferent is the nerve that's mediating the traveling of this input and the afferent sensory input. This is also a nerve. The output motor efferent response is going through a nerve, but it's going to terminate here. It's going to land in the effector. Who is the effector? I just said it. Sweat gland is going to secrete sweat, or your muscle organ is going to shiver. So our effector are always what or what? A gland or an organ. Very good. So our effector is always a gland or an organ. You will see this happening. You will see this loop happening over and over again. What did we mention last week? We mentioned that let's now talk about hyperthermia. It's too hot now. It's too hot now. This is really important. Everybody pay attention. Everybody please pay attention where my talk is. We begin with hyperthermia. It's too hot. Hyper, too hot. Plus, plus, plus. Too much thermia. If there's a hyperthermia, who's going to be the sensor? Is it a photoreceptor, a chemoreceptor, a thermoreceptor, mechanoreceptor, or osmoreceptor? That's a thermoreceptor. The thermoreceptor is going to be sending a input sensory afferent stimulation that it is so hot to the integration processing center. In this case, who is it? Where do we find the thermal nucleus? Where is the thermal nucleus found? You guys did that last semester in the nervous system. The thermal nucleus is found where? In the brain, in the hypothalamus. And then the brain is going to send out an output. What is the output going to say? If the input says it's too hot, what is the output going to say? Let's become even more hot? It's going to say we need to bring the heat down. So who's the effector here? Who's going to bring the heat down? The muscle is going to bring the heat down, says Amapolo. Who's going to bring the heat down? Yeah, very good. That's one response. You're gonna dilate your blood vessels. Remember, we said the ring. Why does the ring feel too tight in the summer? Why does the ring feel so tight in the summer? Because why, if you're wearing a ring, why does it many times when you're so hot in the summer, it feels so tight? Because the blood vessels at the surface of your skin become engorged with blood. Because you're so hot, blood is being shunted to the periphery so that the heat can leave your viscera, your core, your hot core, your intestine, your hot heart, your hot liver, your hot digestive system, so the heat can flow out. That's one response. So here the blood vessel is an organ. The blood vessel is an organ. So when you're hot, the only thing that happens is blood vessels become wide. What's the most obvious one that babies know in the elementary school? When you're too hot, what happens to you? You sweat. So the effector here is going to be the sweat gland and the blood vessels. The sweat gland is going to secrete sweat to cool you off, and the blood vessel is going to dilate to cool you off. Are we clear? Okay, is the sweat gonna happen endlessly? Are you gonna keep sweating on and on and on forever? Or are we gonna stop? Why did we stop? Why did the sweating stop at some point? Why did it not go forever? Yes. Yeah, because we have a temperature that is known as the U range. So you're gonna keep sweating until the hot temperature goes down and down and down. So if you're so hot at 37.9, that's too hot. But if you keep sweating until you return to 37.0 degrees Celsius. When you get there, now you're gonna stop. Now the ring is not going to feel tight anymore because you have reached the euthermia. Are we clear? Okay. We said the opposite. If you're too cold, now let's do the loop again. If you're too cold, who is the sensor? Photoreceptor, chemoreceptor, thermoreceptor, mechanoreceptor, osmoreceptor. That's an example of thermoreceptor. What is the thermoreceptor going to be telling? We have hyper or hypothermia. I cannot hear you. Hypothermia. The hypothermia is going to be carried through an input sensory afferent or through an output motor efferent response. Sensory input afferent response that's going to go to the brain. The brain is going to send out an output motor efferent response to who? To your blood vessel again. To your blood vessel again. But what is it going to tell it? To become wide or become tight now? It's going to make it to become really tight. That's why in the winter, the ring is now going to sometimes feel too loose. Why is the brain telling the blood vessels on the surface feel really tight, to vasoconstriction, in order to conserve the heat, to conserve the too little heat that we have. Imagine if we're cold, we only have just a little bit of heat, and your blood vessels are vasodilating. You're gonna end up losing it. So you need to constrict it to make sure that the heat doesn't escape. Is that the only thing we do when we're too cold? Is the only effector the blood vessels? Who's the second effector when you're too cold? Who's the muscles. What happens to the muscles? The muscles end up shivering. The muscles end up shivering. Excellent. So we have a shivering response. Very good. So now we're up to the pancreas. Remember when we mentioned that some organs are a hybrid, a two-in-one? Remember a hybrid? A hybrid car is like a gas and electric car, two-in-one. Sometimes we have organs that are two-in-one. We discussed that. Remember with the adrenal gland? We said we have an adrenal cortex and an adrenal medulla. If you don't write it, nobody's going to remember it. The adrenal gland is divided into two glands. Two in one. Two in one. There's the adrenal. There's the adrenal cortex. Please write that down. This is really, really important. The adrenal gland is divided into two glands in one. The adrenal cortex and the, what's the second one? The adrenal, remember we said we have a roof and a core. For the brain we say cerebral cortex for the roof, and the core of the brain, what do we call that? The cerebral medulla. For the kidney, we have medullary nephrons in the core and cortical nephron in the roof. So the adrenal gland is divided to adrenal cortex and what else? The adrenal medulla. Very good. The adrenal medulla. The adrenal medulla. Very good. So you also need to know that the adrenal cortex secretes hormones made of what? Secrete hormones that are made of what? This is really, really, really important. Remember we said the adrenal cortex has three zones, the zona glomerulosa, the zona fasciculata, the zona reticularis. So what is the adrenal cortex secreting hormones made of? Cholesterol. So these are cholesterol-based hormones. Every hormone coming out of the adrenal cortex is a cholesterol-based hormone. An example of that would be
Sodium.
example of that, a sodium retention enzyme that is known as aldosterone. Cholesterol, aldosterone, and also we have cortisol, dihydroepiandrosterone. These are all hormones that are made from cholesterol. And there are the adrenal medulla hormones. And who are they made up of? What are the adrenal medulla hormones made up of? The adrenal medulla hormones are made up of what? Adrenal medulla. The adrenal cortex gives us cholesterol-based hormone. If you're made from cholesterol, what does that mean? Does that mean you can dissolve in water? Does cholesterol dissolve in water? Cholesterol is a fat, a lipid, oil. Does it dissolve in water?
No.
So that means all hormones coming from the adrenal cortex will never dissolve in water. And what about the adrenal medulla? They are made up of
Calcium.
cats. Remember when I said catecholamine, catecholamine? You guys remember that from last week? Catecholamines. So the catecholamines, they are polar. The catecholamines are polar, while the cholesterol is non-polar. So you're gonna see the significance of that a little later. There's a significance why we have cholesterol-based hormones that do not dissolve in water. If you don't dissolve in water, what does that mean? Plasma membrane speaking. Plasma membrane wise. The plasma membrane is made up of a phospholipid bilayer. So who's going to be welcome to cross right through the phospholipid bilayer? Is it the cholesterol-based hormone, adrenal cortex hormones, or is it the catecholamine hormones? Who's going to cross right through the plasma membrane? You know, like dissolve like. Likes dissolve like. Dressing dissolved in oil. Glucose dissolved in water. Yes. You had a question? So cholesterol-based hormones, they can cross right through the plasma membrane, while the catecholamines, they are polar. They're not made from cholesterol. They're not made from butter, so they cannot go through the bilayer leaflet. So they need a receptor on the outside. So we're going to be learning that today. That if you're made from cholesterol, you go right through. You have the ID. to go through the cell. Because what are the cell membrane made up of? It's made up of lipids. It's not gonna block you because like dissolve like. You did that in biolab. That things that are, friends of the same feather flock together.
Mm-hmm.
So same thing. If you are made of cholesterol, you're part of the group. But if you're not made from cholesterol, you cannot cross. So you're gonna need a second messenger. A second messenger. Raise your hand if you heard of the second messenger hypothesis. The second messenger. We're gonna see that. Okay, so far, 2-in-1. Adrenal gland is a 2-in-1. It has adrenal cortex and adrenal medulla. What about the pancreas? Is the pancreas a 2-in-1 also? Is the pancreas a 2-in-1? Yes. How is the pancreas 2-in-1? Number one, the pancreas is 2-in-1 because the pancreas is 2-in-1, so we have the pancreas. The pancreas is 2-in-1. Number one, number two. Number one, it's an endocrine organ. Endocrine organ. Number two, it's also an exocrine. It's an exocrine organ. The pancreas is an endocrine organ and an exocrine organ. What does that mean to be an endocrine organ and to be an exocrine organ? What's the significance? The endo, exo. Remember endergonic, exergonic. Remember endothermic, exothermic. So what does that mean? Who want to take a guess? Who want to take a guess for extra credit? By the way, two of you I'm very impressed with. I'm going to give you extra credit. Zaisha and Chanel. They finished all the homework. All the homework up to the exam. That was very, very impressive. Everybody give them a round of applause. I am very happy. So, finish those homeworks as soon as possible and start reviewing them in order to do excellent. If you did not get a chance to finish last week's lab, I'm going to extend it with today. But from now on, please make sure the virtual lab, if you did not get them through, because you didn't have enough time, you didn't have enough time to finish them. So I'm going to extend them and I'm going to make them do with today's lab. Make sure that you finish the virtual labs on time so you can end up with the end of the semester, 3%. Okay? Yes?
So is the homework graded by completion? Is completion right? Or
The homework, no, it's graded by accuracy.
okay.
Yeah. The completion one is the smart book. The smart book assignment, but I don't have those anymore. Okay? All right, endo, exo. And you guys need to review because I'm asking questions that we already discussed in the past lecture and people are blanking on them. They don't remember them. What's that? Yes?
Okay. Can I just take a guess?
Yeah, of course. That's what we need to take an educated guess.
So does it mean that it secretes hormones in the bloodstream?
Yeah, very good. So which one of them? Very good. So bottom line is, the pancreas is going to be secreting hormones. But as Aisha said, we're going to be secreting hormones either into the blood, like insulin. That one needs to go in the blood to lower down blood sugar. So it's got to be in the blood to bring down blood sugar. The police need to be where crime is to bring down crime. And the exocrine means what? Yes. Yeah, so it's excreted extracellularly, not in the blood, but into a tunnel known as duct, into a duct. So if you're secreted into the blood, that means it's an endocrine hormone. But if it's secreted into a duct, that's an exocrine hormone. An exocrine hormone. Are we clear? Okay, so endocrine is the blood, exocrine in the duct. Now, for the endocrine, you need to know insulin and glucagon. This is so important. Insulin and glucagon. Everybody, can I have your attention here, please? Let's begin with insulin. When does insulin work? When we have too much sugar. What do we call too much sugar? What's sugar? We don't say hypersugaremia. We don't say hyposugaremia. We say glycemia. You know, we have our own language. So, when is insulin secreted? When we have hyperglycemia or when we have hypoglycemia? When is insulin secreted? When you have too much blood sugar or too little blood sugar? Insulin. When is insulin? Yes. When you have too much blood sugar, when you have hyperglycemia, when the sugar is too high, you had a very big meal, especially when you eat in IHOP where they have all the carbohydrates and the pancakes, those hot fluffy pancakes. Anyways, so the hyperglycemia is when there's so much sugar. If there's so much sugar in the blood, that leads to coma. So we need to get rid of the sugar in the blood and send it where? Inside the cell. So who's gonna do that? It's insulin. So insulin will come out from when there is hyperglycemia. But let's now go through the loop again. Like I said, this loop is gonna be going over and over for all the different hormones. So who's the sensor who's gonna detect hyperglycemia? It's too much sugar. Is it a photoreceptor, chemoreceptor, thermoreceptor, mechanoreceptor, osmoreceptor?
Mechanoreceptor.
Is it a mechanoreceptor? Are we sensing mechanical sensation, distension, vasodilation, vasoconstriction?
I meant chemo.
Chemo, very good. So that's because glucose is chemo, that's a chemical. So hyperglycemia, then the receptor is found where? In the pancreas. The pancreas is gonna say, the pancreas is like tasting the blood. Oh my God, the sugar in the blood is too hot. That is a receptor, a sensor of the blood in the pancreas. Then the pancreas itself, it's gonna send the sensory input to the endocrine pancreas or to the exocrine pancreas now. If the blood sugar is too high, is this going to be going to the endocrine pancreas or the exocrine pancreas? The endocrine pancreas. Then the endocrine pancreas is going to cause the release. So the effector is still, you see the pancreas is 3 in 1. It was the receptor who detected that's too much sugar. It was the integrating center who decided to secrete, because there's two options, insulin and glucagon. So it secreted insulin. And lastly, it was the effector who actually squeezed out the insulin, and insulin brought down blood glucose. So, and then is glucose going to be secreted forever?
No.
Imagine you secrete too much glucose, now you're going to drop from too much sugar to too low sugar, which is another problem that leads to coma. So you need to come down and stop. What is that called? When you just fix it, just remember last week when we were saying the speed limit, you press your brakes, and you're right at the speed limit. You don't go below the speed limit, you don't go higher than the speed limit. You maintain the homeostasis. So now what is that called? Euglycemia. That's called negative feedback. The opposite, if you have too little sugar, hypoglycemia. Who are we going to secrete now? If there's too little sugar, who are we going to secrete now?
Glucagon.
We're going to secrete glucagon. What is glucagon going to do? It's going to start depleting, bringing out, liberating glucose from the glycogen. And then that's going to replenish. It's going to replenish the glucose supply in the blood. Are we clear? So that's endocrine, hormone, insulin, glucagon. What about exocrine? Exocrine, that's for the digestive system. You see? Look where my mouse is. You see? This is, what is that? Does that look like a blood vessel? Please look here. Does that look like a blood vessel? We did that in biolab. My biolab students. You guys remember that. Remember when we did the duodenum? What is that? That's the duodenum. So the pancreas is right at the duodenum. Remember when we said endoscopy is where you put the camera through the oral cavity and you end at the duodenum. And colonoscopy is when you enter through the rectum and you end still at the duodenum. So this duodenum has a neck. Why does it have a neck? Because the head of the pancreas is pushing against it. Why does it push against it? You see this tunnel? Do you see the tunnel? Ariana's friend. You see the tunnel I was saying earlier? The tunnel, you squeeze the exocrine juices, the hormones, into a tunnel known as what? Duct. You see the duct now? Because the duct is, what is the pancreas secreting? The pancreas is secreting pancreatic lipase. What do you think that would do? Pancreatic lipase.
It breaks down the lipids.
Is gonna break down the lipids. Very good. Pancreatic amylase. What is that gonna break down?
It's gonna break down the amino acids.
It's gonna break down the sugar. Amylase. Amylase breaks down the sugar. Who breaks down the proteins? Who breaks down the proteins? Pancreatic.
Something.
Peptidase. Peptidase. Pancreatic peptidase. So you need to know who's endocrine, who's exocrine. You can't say that insulin is an exocrine hormone, or insulin is pancreatic lipase is an endocrine hormone. No, pancreatic lipase is exocrine. Are we clear? Okay, so now let's go into the slides. Glucagon. Glucagon is secreted by the alpha. Glucagon is secreted by alpha cells. It's released between meals when the blood glucose is falling. You see? Falling. That's the key term. Falling. What is blood sugar falling called? What do we call that? What do we call blood sugar drop? Blood sugar. You see, these are the words that I showed you there. We have arrows there. Please put away the phone. Arrows there, pointing up, it means increase. Arrows going down means decrease. So when we say falling, does that sound like a problem or no? That's a hypo. Very good. So a hypo what? Glycemia. So there we go. Glucagon comes out when there is a hypoglycemia. We need to put it up. It was released between meals when blood glucose concentration is falling. So in the liver, it stimulates gluconeogenesis. You know genesis? Genesis means the making. So gluco, meaning glucose. Gluco means glucose. So what is gluconeogenesis? What's this neo? You know neo? Neo-Nazis? Neo means new. So what does that mean? It doesn't mean there's new Nazis. It means that we are making new glucose from what? From non-glucose precursors. Okay? It means that gluconeogenesis means when we make glucose, okay, everybody pay attention. If you make glucose from galactose, is that magic? That's normal because galactose is a sugar. When you make sugar from a sugar, is not something cool. If you make burger from beef, is not cool. It's when you make a veggie burger. That one is cool because you made it from different non-traditional, non-classic ingredients. That's the same thing for gluconeogenesis. Gluconeogenesis is not when you make glucose from fructose. It's not when you make glucose from sucrose. It's not when you make glucose from galactose. It's not when you make glucose from maltose. It's when you make glucose from proteins. And that happens when there's a poverty. When there is very, very bad poverty and famine and people cannot eat, they don't have enough food, what happens? The body starts to break down the protein in the muscle. So when you make sugar from protein, is that Now, new making of sugar. True or false? Yes, it is. Because you're not making sugar from sugar. You're making sugar from protein. That's gluconeogenesis. You need to know what gluconeogenesis is. What's glycogenolysis? So break down the term. You know, in the spelling bee, they say, what's the origin of the word? What's the blah, blah, blah. You know, they ask these kind of questions. That's actually very interesting. Because if you look at a word like gluconeogenesis, you can define it if you break down the words. Gluco means glucose. Neo means new. Genesis, making. It's the making of glucose from new sources. Not from galactose, not from maltose, not from sucrose, not from fructose, but from what? From proteins. Who can define what gluconeogenesis is? Yes?
Breakdown of glycogen into glucose molecules.
That's glycogenolysis. Yes, so
The making
lysis,
of the glucose.
breakdown of glycogen. So to make glucose from glycogen, to break down glycogen into glucose. But what is gluconeogenesis?
The making of the glucose in the liver.
The making of glucose from non-glucose precursors, non-glucose ingredients. It's like when you make a veggie burger. You made a burger, but not from beef, not from chicken. So you made it from veggies. So that's the new burgerogenesis. Okay? In adipose tissue, what kind of tissue is that? Is that a nerve tissue? Adipose tissue? Is that a nerve tissue? That is a fat tissue. Excellent. Very good. And it stimulates fat catabolism and release of free fatty acids. What is that slide showing you? The slide is showing you there the effects of glucagon on all of these different effectors. These are effectors. So the effect of the liver, it makes it secrete glucose. The effect on the adipose tissue, it breaks down the fat. So that makes sense. The effects of glucagon on the protein, it breaks down proteins. So that makes perfect sense. Why does it make sense? Because this is a glucagon that's trying to give us energy or is trying to save energy. What is glucagon doing? Is glucagon a energy saving hormone or an energy consumption hormone? Glucagon. Think about it. Glucagon makes glucose come out. Does that mean it's going to be burning glucose or saving glucose? It's going to be burning glucose. Excellent. So is glucagon catabolic or anabolic hormone?
Wouldn't it be saving it? Because if we don't have any, if we're in hypoglycemia, we don't have any. So shouldn't we be conserving it?
No, the one conserving it is insulin because you have too much IHOP. You ate too much IHOP and there's so much sugar. Too much. So then you only save when there's too much. But in glucagon, there's too little. So we need to bring out from the liver. Where is glucose found? In the liver and in the muscle. In the liver and in the muscle. Yes, Ariana?
I was going to say it was a catabolic process.
It's a catabolic process. Excellent. Because we're breaking down. We're catabolizing. But insulin now is an anabolic hormone. And that is another essential hormone. Insulin is a very essential hormone. People that have a lack of insulin have an essential disease, an essential clinical problem, like diabetes type 1 and diabetes type 2. So, insulin is secreted when you're hungry or when you're full?
When you're full.
When you're full. When you ate the IHOP hot and fluffy pancakes with the syrup. That one, you have too much glucose in the blood, and you need to be saving the glucose. Somatostatin is secreted by, so you remember, we're studying two, not just the physiology, but also the anatomy. So don't just remember that insulin is a anabolic hormone, glucagon is a catabolic hormone, but remember where it's coming from. Don't just say the pancreas, because the pancreas has different boroughs, like New York City has Brooklyn, Queens, Staten Island. The pancreas has alpha cells, beta cells, delta cells, gamma cells. You need to know where exactly it's coming out from. Just knowing the insulin is not enough. That's layman in the street knows that. People who watch Good Morning America, they know that insulin comes out the pancreas. But you're taking AMP, you need to know where exactly it's coming out of. So insulin is coming out from where? Beta. That's the beta cells. Glucagon is coming out of where? Alpha cells. Somatostatin is secreted by what? Delta cell. So this is showing you a histological section of the exocrine and endocrine pancreas. Now we're going to talk about the gonads. What did we say that was so interesting about the gonads? You remember our axis? Remember our axis? In our axis, remember the treasure map I drew last week? Who remember the treasure map? Remember the treasure map that we drew last week? When we had an A, B, C, and D? That was a treasure map. You guys remember that one? A, B, C, and D?
Oh,
We have A. Who is A? Very good. So the hypothalamus is the one at the top. The hypothalamus. Then the hypothalamus tells B. Who's B? The pituitary gland. And who is C?
target cells.
C is the target cell.
No, target glands.
Yeah, the target gland. Yeah. Remember we said, I'm a Polo. Remember we said that in the endocrine system we have three different effectors. We have the glands, we have the organs, and we have the cells. So in this case, since we're going to talk about the gonads, we're going to talk about a target organ. Okay. What did I say that was so interesting? Remember last week I said if you put water in a strawberry plant, it's going to give you a strawberry. You put water in an orange plant, it's going to give you oranges. You put water in banana plant, it's going to give you a banana. Even though it's the same water. H2O. Same thing with the hormones. Remember what I said last week? That the hypothalamus secretes hormones that end in what? There is a suffix. If it ends with that, that means it's coming from the hypothalamus. What is it called? Releasing hormones. Excellent. So we have GnRH, gonadotropin releasing hormone. Gonadotropin releasing hormone is going to target the pituitary gland. Then the pituitary gland is going to send not one, but actually it's going to send two. It's going to send two hormones. The first one is called follicle or follicular stimulating hormone. And the second one is called what? Excellent. It's called the LH or the luteinizing hormone. And they will get to the target organ. If the target organ happens to be testes, you will get sperm and you will get what else? Eggs in the testes.
Testosterone.
You're going to get testosterone. You're going to get sperm and testosterone. But if this axis here, this axis is found in both men and women, in males and females. The only problem is the C part. A and B are the same. So you have GnRH for men, GnRH for women. You have FSH and LH for men, FSH and LH for women. But when the FSH and the LH arrive at the ovary, what will they give you? They will give you the ova, the eggs, and what else will they give you?
The estrogen.
They will give you estrogen. They will give you estrogen and they will give you an ova. But if you see them the same ones, FSH and LH, on the testes they will give you sperm and testosterone. Remember what I said about water? You put water in banana plant, it will give you banana. Put water in mango, it will give you mango. Even though it's the same water. Same thing here. FSH and LH are the same FSH and LH. You put them in the ovary, you get ova and estrogen. You put them in testosterone, you get testes and you get sperm and
Testosterone.
testosterone. So the gonads are the testes and the ovaries. The ovaries for the female. You have theca cells that synthesize androstenedione. It is converted to mainly estradiol by the granulosa cells, and after the ovulation, the remains of the follicle becomes the corpus luteum. It secretes progesterone for 12 days following the ovulation, and the follicle and the corpus luteum secrete inhibit. So the functions of estradiol and progesterone. Progesterone, what do you think is that made up of? Protein, catecholamine, or do you think progesterone is made up of nonpolar cholesterol? What do you think progesterone is made up of? What kind of hormone is progesterone? Is it protein-based hormone, or is progesterone cholesterol-based hormone?
Protein.
It ends with sterone, like aldosterone, like cortisol, like estrogen, testosterone, progesterone. They all end with rone because that means what? They're all made up of cholesterol. Are we clear? Are we all clear? Okay, we talked about the okay, vitamin D. How do we get vitamin D in the blood? Vitamin D is important because it is responsible for what? What is vitamin D responsible for? It's responsible for absorbing calcium from the intestine into the blood. So you can drink all the milk you want with the cereal, Fruity Pebbles, cereal, and milk, but it's never going to be beneficial unless you have vitamin D. Does that make sense? Are we all clear? If you drink all the milk in the world and you have low vitamin D, the milk is going to stay in your digestive system until it gets excreted. It will only get absorbed if you have the proper amounts of
Vitamin D.
vitamin D. Are you guys following? What's the significance of vitamin D? Vitamin D is responsible for what? Vitamin D is responsible for the absorption of calcium from where? The absorption of calcium from the, yes, from the intestine when you drink milk into where? Into the blood. And then the blood is going to deliver the calcium into the tissue, to the tissue compartment, so your tissue can finally use it. Very good. Excellent. Excellent job. So the kidneys. The kidneys are important for the release of 3 hormones. Number 1, calcitriol. Calcitriol is the bioavailable and and active form of vitamin D. It increases calcium absorption by the intestine and inhibits the loss in the urine. The kidneys secrete renin that converts angiotensinogen to angiotensin 1. So now we need to talk about the renin angiotensin aldosterone axis. The renin angiotensin aldosterone axis. And we will also talk about the EPO. Okay, so if we look into the kidneys are responsible for converting the calcidiol into calcitriol, converting the vitamin D from an inactive form into an active form. And that is gonna release calcium from the intestine, the calcium you just had when you drank the milk. Now it's going to be flowing into the bloodstream and from the bloodstream into the tissue. The kidneys are also responsible for secreting renin. So this is known as the RAS system, R-A-A-S. What is RAS? RAS is the renin, angiotensin, aldosterone axis or system. So what happens is whenever there is a drop in blood pressure, whenever there's a drop in blood pressure, is that clear? If there's a drop in blood pressure, what is that? Hyper or hypo? Drop in blood pressure. Is that hyper or hypo? That is hypo. A drop in blood pressure is hypo what? That is hypotension. That is hypotension. Whenever there's hypotension, the kidneys, your kidneys, people think kidneys are only good enough for secreting urine. So the kidneys are gonna secrete, the kidneys are going to secrete renin. What is renin gonna do? Renin is an enzyme that's gonna convert angiotensinogen. Nogen means inactive into angiotensin. That's the active form. Angiotensin. So renin is gonna convert angiotensinogen into angiotensin. Angiotensin is going to go in the lungs. And there's an enzyme waiting there called ACE. Anybody heard of ACE inhibitors? ACE inhibitors. So ACE is an angiotensin converting enzyme. So we're going to take angiotensin 1 and we're going to convert it into angiotensin 2. Angiotensin 2 is going to have effects that are going to restore the drop. So our problem was there was a drop in blood pressure. There was a hypotension, wasn't there? There was a hypotension. So renin is going to cause angiotensin 2 to be secreted terminally, and angiotensin 2 is going to restore the blood pressure back. It's going to return blood pressure back to normal. Are we clear? Okay, we have EPO now. So that was RAS. You need to know both RAS and EPO. What does EPO do? EPO stands for erythropoietin. What does erythropoietin mean? From the word erythro. You can tell what that is. What's erythro?
Red blood cells.
Yeah, excellent. Erythro is red blood cell. Very good. What is poietin? Poietin means it makes red blood cells. So if you look at this loop that we have, when we looked at the renin angiotensin aldosterone, What was the problem? Hypertension or hypotension?
Hypo.
There was a hypotension. Who sensed it? Mechanoreceptor. The mechanoreceptor sent the input sensory signal, and the sensory signal went to the kidney, and from the kidney into the lungs, and then we had an integration and an output that caused the blood vessels to dilate and led in the end to an increase to restore the blood pressure. Correct? Now, if you look at EPO doesn't do that. EPO does not sense blood pressure. EPO does not sense temperature. EPO senses oxygen. So it's tasting the blood for the amount of oxygen. If the amount of oxygen drops, what is that called now? Hypooxygenemia? No, that's called hypoxemia. Hypoxemia. So when oxygen drops, not blood pressure drop, blood pressure is okay. When the oxygen drop, what is that called? When the oxygen drops in the blood, what is that called?
Hypoxemia.
Hypoxemia. Very good. Hypoxemia. So is that a problem or no? Hypoxemia. That's a problem. What do we need to restore hypoxemia into? We're going to restore hypoxemia into
Eoxemia.
euoxemia. Very good. We need to restore it to euoxemia. We need to restore it into euoxemia. The hypoxemia is going to send the input. The input is going to go to the integration center, and the integration center is going to be your kidneys. So now when there is low oxygen, who carries oxygen in the blood? You just said it. Red blood cell. So what does EPO do? EPO is going to increase the making of red blood cell. This point is really, really important. What does EPO do? EPO is secreted when there is low blood oxygen, and it's going to increase the amount of red blood cells to help accommodate more oxygen, to replenish the oxygen. Yeah.
Does that have to do with like anemia?
Yeah, so anemia is when there is a complete drop in red blood cell count. Yes, so when there is low blood cell count, your EPO is going to be secreted to replenish it. But if there is not enough iron, because you know red blood cells are made up of hemoglobin, heme has iron, so you need to ensure you have the proper iron supply. So females in the reproductive age, due to the menstrual cycle, lose a lot of blood and lose a lot of iron. So they need to take the multivitamin pill. And if the girl or the young female in the reproductive age does not take the pill, what's going to happen? She's going to have anemia, and she's going to be so pale and so dehydrated and very weak. Why? Because she's losing a lot of blood in the menstrual cycle and not taking enough iron to replenish that supply. Are we clear? Okay. The heart. Remember I said earlier that the heart is very interesting because it secretes a very interesting hormone known as atrial natriuretic peptide. So the atrial muscle secretes two natriuretic peptides in response to an increase in blood pressure. So the heart can respond to high blood pressures, and it can bring the blood pressure down. Adipose tissue secretes leptin, and the leptin is going to slow the appetite because you already have too much fat tissue. You don't want to, if you have already too much tissue, you do not want you do not want to increase any more amount of fat, because that's going to lead to atherosclerosis. The placenta is going to secrete estrogen and progesterone to help the development of this embryo that implanted into uterus. And we talked about that earlier, that we have two kinds of hormones. One is made from what, and the other one is made from what? The two hormones. We have two classes of hormones. One class of hormone is made up of cholesterol, and the second hormone is made up of estrogen. One class of hormones is made up of steroid, and the other class of hormones is made up of proteins. So what's the significance? Okay, so now we're finished with this loop, with sensory input, sensor receptor, sensory input, afferent stimulation, integration, output, and what comes after the output is the effector, who's going to be doing the final response. Is that clear? Are we clear? Okay, so if you now look at the hormones, what are the different types? What are the different types of hormones structurally? Are all hormones the same? Chemically, structurally? Are all hormones the same? Are you guys following? Are all hormones the same? Before we start the lab, I just want to go over this very important point. Now, hormones are not all the same. So, the structure of the hormone. So, hormones structurally are classified. This is so important. Please pay attention. Hormones are divided into two categories. Category number 1 and category number 2. Category number 1 is known as non-polar cholesterol based hormones. And then there is the other category known as the polar, non-cholesterol based catecholamines. So one is polar, one is non-polar. Are we clear? One set of hormones is polar, while the other set is non-polar. The set of non-polar cholesterol-based hormones, the set of non-polar cholesterol-based hormones. What does that mean? Let's look now at the structure of the cell. We know already that the cell membrane has a bilayer phospholipid bilayer. The core is what? Is non-polar. The middle part, the middle here, this middle part, excellent. I found something orange. Orange like a butter. Think of this like butter. This is non-polar. This is non-polar. Non-polar. But what about the tips here? Good, I found red. So that is what? Red is what? Red means that this is polar or non-polar? So the orange part is nonpolar, and the red part is. So that's the cell we're trying to get into. Now, if you look at our let's also make our two hormones, one nonpolar in orange, and one polar in what color?
Red.
Red. Please look at that cell membrane. Who do you think is going to penetrate and cross into that cell membrane? The orange nonpolar or the red polar? The orange one. Very good. Not only it's going to cross, like let's say this is the cholesterol-based hormone, like testosterone, progesterone, aldosterone. They will cross the bilayer leaflet. They will bind cytoplasmic receptors, and not only that, they can go where? They can go where? What is this I'm drawing now?
The nucleus.
I'm sorry? Be nice and loud. What am I drawing now? What is this thing that I'm drawing now? That has these holes. What am I drawing now? This purple thing that has holes in it. It's a purple thing that has holes in it, and it has a lot of
The nucleus.
that's the nucleus. So now this particular hormone that is nonpolar is gonna bind to the receptor, and it's gonna mobilize to the inside, and it's gonna bind to the DNA, and it's gonna transcribe for us. It's gonna transcribe for us DNA, and it's gonna give us an mRNA. The mRNA is gonna be translated in the ribosome. Is going to be translated in the ribosome to give you your target, protein. And your target protein is going to give you your target response. So you see how nonpolar hormones work? They cross the bilayer leaflet, they cross the nucleus, and they transcribe DNA to give you mRNA. The mRNA is going to be translated in the ribosome to give you your protein. That's going to do the job. But what about the red ones now? These polar catecholamines. Can they cross? Can they cross now? They cannot cross. So what's going to happen? They cannot do their job now?
No.
So how do we solve this problem? Let's say you call an Uber. And the Uber student is just like the Uber who's going to pick you up, happens to coincidentally be an LIU student. They can walk right into the school, right? And pick you up, for example. But if you have an Uber driver who cannot get in the school, so what are we going to do now? You're going to need, they're going to talk to somebody who can go in the school, and that somebody's going to get to you. So we're going to need a second messenger. So now the red stuff is going to bind the receptor on the surface. So this red polar hormone, what's an example of that? Those are all like epinephrine. That's an example of a red polar. So these cholesterol-based hormones are testosterone, progesterone, estrogen, cortisol, aldosterone, and so on. But the red one, an example of that is epinephrine. Once epinephrine binds to its receptor, it cannot get in. So it binds to a receptor facing on the outside. Once epinephrine binds to a receptor facing on the outside, What is epinephrine going to do now? It's going to stimulate a second messenger. A second messenger. A second messenger called a G-coupled protein. This is known as a G-coupled protein receptor. This G-coupled protein receptor is going to go to another protein that is here on the plasma membrane known as adenylyl cyclase. Adenylyl cyclase. What does adenylyl cyclase do? It's going to convert ATP. Everybody knows ATP. Everybody knows ATP. It's going to convert ATP into cyclic adenine monophosphate. Cyclic AMP. Cyclic AMP is my second messenger. That's the public safety guy who's going to walk in and tell you your parents are outside or your Uber driver's here. That's the second messenger. So cyclic AMP is the second messenger. It's going to go and it's going to do the target effect by stimulating some kinases. Some kinases. What are kinases? Kinases. What are kinases? Kinases are enzymes that add a phosphate group. Kinases are enzymes that add a phosphate group, and once they add a phosphate group to the enzyme, they're going to activate the enzyme. And then the enzyme is going to give you the target response. So compare the two. The cholesterol base went right into the nucleus to give you the protein, to give you the target response. But the epinephrine, the polar one, cannot come in because it's polar. It has to stimulate the cyclic AMP in order to activate a kinase. Then the kinase is going to activate the target enzyme. This is really important. You guys see this in pharmacology a lot. Many medications, stimulate these second messenger pathways. Are we clear? Okay. So if we look there, all steroid hormones are cholesterol based. All steroid hormones are cholesterol based, and they are derived from what? Cholesterol based, and they are derived from cholesterol. Example of that would be the sex steroids like estrogen and the corticosteroids like cortisol. We discussed that. The catecholamines or the monoamines, these are your dopamine, epinephrine, norepinephrine, melatonin, thyroid hormone. These are all cannot cross the lipid bilayer. You see now, these are the steroid hormones. They have 4 rings. Always, when you see 4 rings, you remember that what is that now? If you see 4 rings, what is that?
Cholesterol.
That means this is cholesterol. It means you're made up of cholesterol. So, does this dissolve in water? Does not. What about here? Does that dissolve in water now? Absolutely. So peptides are created from multiple chains of amino acids. If you look at this, does that look like a cholesterol-based hormone, or does that look like that doesn't look like a cholesterol-based hormone? Does that look like a protein-based hormone? This here. Is this cholesterol-based? If you look here, you have peptides. Peptides. Insulin is a large peptide hormone. It's made up of peptides. Oxytocin is also a peptide hormone. But if you look at that, is this peptide? This has how many rings?
4.
Now you see how they're all made up of cholesterol? Who did we say has a 4-ring structure? Cholesterol. So they all have cholesterol. So that's cholesterol. This is progesterone. This is testosterone, cortisol, aldosterone, estradiol. They're all made up of cholesterol. What is this now? You need to know this structure. This is the structure of, that is the structure of insulin. So insulin, is insulin a peptide hormone, or is it a polar? Is it a polar? Is it a cholesterol-based steroid hormone, or is it a peptide catecholamine hormone? It is a peptide. So does insulin bind on the outside because it cannot come into the cell, or does it cross right through the membrane, the plasma membrane? Please raise your hand. Insulin. Is insulin a cholesterol-based hormone that can cross the plasma membrane, or is insulin not a cholesterol-based hormone and a peptide hormone that needs a second messenger? Yes.
They cannot cross the membrane.
It does not cross the plasma membrane, and it needs therefore a second messenger. Okay, so let us stop here. Everybody, please take out your lab worksheets and assemble into groups, and work together to answer all the questions that are in the lab worksheet. If you go online, you'll be able to find that I have also for you, I have extended the virtual labs from last meeting if you missed them, because that was the first meeting. And today we have a dissection that is also on the McGraw-Hill. Please log into McGraw-Hill also, and you will be able to see that we have a dissection. Yes.
For the endocrine system APR, it's still locked.
Yeah, that one I'm gonna open it now.
So do we need to also get that done, or do you just want us to do the worksheet?
Yeah, both. So you have to finish both within by the end of the class.